Angie Liew, SBI4U Period 1. The purpose of this experiment done by the students was to discover the effects of different sugars on anaerobic cellular respiration of yeast. The hypothesis was that glucose would have the highest amount of bubbles, maltose would have the second highest, and sucrose would have the third highest, because glucose is already a monosaccharide and can be easily used in glycolysis. Maltose contains two glucose molecules but will take more effort and energy to break down, and sucrose contains less sugar than maltose and would take more effort to break apart. The process includes setting up the experiment, leaving the boiling tubes in the water bath and waiting for oxygen deprivation, recording the results, and analyzing results. The independent variable would be the type of sugar used. In this case, it's glucose, sucrose, and maltose. The dependent variable would be the rate of anaerobic respiration in number of bubbles per minute. The bubbles are carbon dioxide, which are a measure of rate of respiration because ethanol fermentation produces carbon dioxide. The oil layer is to prevent oxygen from entering the yeast-sugar mixture, and the constant control of the water bath at 35 degrees is just kept constant throughout the entire test. The overall results, after 2 trials, glucose produced 26.5 bubbles after 2 minutes, sucrose produced 22 bubbles after 2 minutes, and maltose produced none. Possible interpretations by the students would be that glucose had the most amount of bubbles because it was easily used by yeast for glycolysis. Maltose had no bubbles, possibly because of an error in equipment and preparation, or that yeast could not break down the maltose glycosidic bonds. Sucrose had the second highest amount of bubbles because fructose might have not been as efficient in cellular respiration as glucose. These are the possible sources of error. The yeast might not have had enough time to become active. It might have been contaminated, there might have been a human counting error, the yeast died since the 2 trials were held hours apart, or that the bungs were not tightly placed and gas might have been able to leak out. These are the expected results according to research. Sucrose was supposed to have the highest amount of bubbles, glucose would be second highest, and maltose in third place. In conclusion, the students found that glucose produced the most bubbles, sucrose produced the second most, and maltose produced no bubbles. However, these results were unexpected and contrasted research done by other sources.